let me introduce Dr. Mathieu Groussin from Kiel University in Germany with the talk now, the Microbiome Conservancy, Preservation and Multiomics Investigation of the Global Gut Microbiome. Please, Mathieu. I will let you know before 2 minutes before the end of the talk.
Sure, thanks. Can you all see my slides in presentation mode, or do you see like
no, we see the viewer from the speaker.
Okay. So
yeah,
swap display. Is that okay?
That's okay.
Okay, great, thanks. So sincere apologies for this confusion. I'm sure I'll be the only one in the whole conference mixing it up. So I'm very ashamed of being unique here. However, I'm very happy to present. Thanks again for inviting me. I'm going to talk about the research and the work we've been doing with the Global Microbiome Conservancy that we started while we were postdocs at MIT in the lab of Eric Kalm, and that we are continuing doing now as independent group leaders at Kiel University in close collaboration with Thomas and Thomas Bosch that is also in the audience. So I'm sure that there's no need to introduce here why it's important to study the microbiome of underrepresented populations, especially in the context of global industrialization and urbanization of the world that associates very strongly with a rise in microbiome-associated chronic disease. And so as we better understand the mechanisms and the drivers of such disease, we came to fully and more importantly appreciate how much the microbiome plays a role here and is affected by not only the changes in the lifestyle, but by these diseases. And so better understanding the complex relationship between these tripartite players, industrialization or lifestyle, non-communicable disease and host microbiome system is an urgent imperative for all of us, which motivated us to actually create this effort, the Global Microbiome Conservancy, which is a non-profit initiative that aims at preserving and understanding the diversity of human microbiomes worldwide. And so we have 4 core activities which revolves around conservation or preservation of the microbiome, as well as a representation of human populations that have diverse genetic backgrounds, as well as differentiated lifestyles. We also obviously generate data and science to advance knowledge and how the microbiome is impacted by these lifestyles and the rise of chronic disease. And we also engaged in capacity building with the collaborators we've been working with in different countries, and that I will go back, I'll circle back on this a bit later. So in terms of capacity building, despite being a young initiative, what are we, for instance, proposing? Because this is really key in our activities. We are now currently developing a model where we can propose funding and grants to local investigators to lead microbiome research in partnership with the Global Microbiome Conservancy, so they can submit a proposal and receive a grant in like several dozens of US dollars. But we also very soon in 2024 start and organize an in-person bioinformatic workshop here in Kiel at Kiel University, where students of our PIs and our collaborators who are part of the GMBC, Global Microbiome Conservancy Consortium, will be invited to come in order to do bioinformatic training on microbiome analysis. And so far, we engaged in collaborations across dozens of different countries and multiple collaborators. As highlighted here, we recruited a cohort of about more than 1,000 participants, and we're able to culture gut microbes, because we're focusing on the gut at the moment, from healthy populations, to culture diverse gut bacteria that we biobanked in freezers in order to promote dissemination of this resource as well as investigation on this resource. So what about the cohort 1.0, I should say, comprising the first set of localities and countries that we visited ourselves and where we recruited individual cohorts. So we recruited individuals in urban localities as well as rural localities in the different countries across several continents. And from the samples we collected, we generated genotyping data for getting human genetic information, as well as microbiome data in the form of sequencing. So we sequenced the metagenome, but we also spent a lot of effort and a lot of time in generating biobanks of isolated strains. With the cohorts that we recruited, we also collected a wide range of lifestyle and dietary metadata that I present very briefly here in the bottom panels, where you can appreciate the diversity that exists in terms of lifestyle factors or dietary factors across the cohorts. And this was intentional because we really wanted to start to tease apart the contribution of lifestyle versus diet versus host genetics on the distribution and shaping the profiles of microbial diversities worldwide. This is just to illustrate the diversity of admixture groups, of genetic populations that we have in our cohort. In the first cohort, and the people can be separated into like about 17 different admixture clusters that we can then use to correlate with the microbiome data. What does the microbiome diversity or alpha diversity look like? As you can see here in this plot, we can clearly see that the countries, the people from countries living more industrialized and urban lifestyles have diminished microbial diversity compared to populations on the left who are more rural and living in non-industrialized places. These correlate not only to just to the first dimension of the multivariate analysis of lifestyle factors, but also very much to the locality density of the different populations. And so in multivariate models, we can now account for multiple factors, including environment, including genetics, including lifestyle or diet. And we can then now measure how much the diversity or how much the variance in alpha diversity across populations is driven or associated by each of these individual factors. And we start to now realize how much each of those really contribute significantly individually to the differences in alpha diversity between populations. We can also use the same approach in order to understand the patterns of beta diversity, so the differences in global microbiome compositions between human populations. There's a clear shift that associate in microbiome compositions that associate with urbanization or locality density. But actually, when we measure the contribution and we do like variance partitioning and measure the contribution of the different factors in driving the differences in microbiome compositions between people, we're able to measure significant associations with all of these different factors and still realize that how much the correlation between lifestyle, diet, and host genetics on its own is also associated with microbiome compositions, and it's still quite hard to fully disentangle the contribution of each of those factors. However, one thing that is interesting is that rather than looking on the left here at how much these different factors associate with global compositions, we can also look at the distribution of individual bacterial taxa and how much do they correlate with different host factors. And in past studies, certain bacteria in the gut were highlighted and found to be strongly associated with lifestyle and the level of industrialization of the countries where people were coming from, and were called key markers of lifestyle and industrialization. But now in a multivariate model where we can also account for the contribution of host genetics and diet and geography and other confounders, we realize that these associations that we find very strong and very significant here in full dots with just lifestyle or industrialization, many of them get non-significant when you actually account for the other contributors such as genetics and diets, which now also helps really identifying the individual bacterial taxa that are really more markers of lifestyles. What we did with the microbiome data is ask the question of whether, in terms of co-abundance profiles in microbiome networks, we can identify differences in the structure of the networks between what we could call an industrialized versus a non-industrialized gut microbiome. And when doing so, controlling for the differences in alpha diversities between our groups, or the differences in localities, we can observe that there is a decrease in the degree, and so in the connectivity of the bacteria in the networks of industrialized microbiomes, which are also less stable than non-industrialized microbiomes. And now the last point that I wanted to discuss and present briefly today is again illustrating how much it is important to characterize the microbiome of underrepresented populations, because when looking at the gut microbiome of industrialized nations, we thought that we could actually find ways to build novel biomarkers of chronic disease by looking at the shifts in microbiome compositions between health and patients. And such biomarkers or such indexes were built. For instance, this one, the Gut Microbiome Health Index, which was proposed in 2020 by Gupta et al., looked at the distribution of 50 marker species and could build an index based on the distribution of these key species that was very powerful in discriminating between healthy people and people affected by several types of chronic disease. And what we wanted to test is whether this gut microbiome health index was also capable of differentiating health versus disease in our cohorts, considering that all of our individuals in our cohort were screened for being healthy. And what we realized is that the index was quite powerful in identifying people who are healthy indeed in Canada, Finland, and the United States, as expected. But for most of the countries, all the admixture clusters that we sampled, we realized that this gut microbiome health index, at least the median of it, was falling to 0, and we had no real capacity to identify whether or to call whether a person was healthy or disease based on the microbiome profile, simply because, as you can see on the right, there is basically no of such key health marker taxa in the non-industrialized populations, really suggesting that such indexes or such biomarkers that we build and define from the microbiome data of industrialized nations have very low portability when it comes to other populations of the world, which is also mirroring pretty much what we observed by looking at biomarkers that use genomic data, such as polygenic risk scores. So with the samples, as I said, we in the field, we also preserve the samples in such a way that we can do anaerobic culturomics later in the lab, which we did. And so we used the culturomic approach to generate a wide range of to generate a collection, of diverse bacterial species. We have about like 500 different bacterial species now in the lab that contain about 10,000 different strains in total. In this particular paper in 2021, we could show that because we generated the whole genome of each of those isolates, we could now have a better representativity of people outside the industrialized and urban world in the collection, which is nice because now we can have bacterial genomes that associate with a wider range of host lifestyles. And we leveraged this collection of genomes to actually investigate more particularly patterns of horizontal gene transfers in the microbiome. And we were very happy to see that actually these patterns, as the frequency of gene transfers in the microbiome, as well as the functional profiles of genes that were exchanged, were very different between the microbiome of industrialized populations compared to non-industrialized microbiomes. And what was interesting is that we were able to measure an elevated rate of gene transfers for bacteria that occur in the industrialized microbiome compared to non-industrialized ones. So how much does that impact or connect to host physiology or human health, and the chronic disease we see in the industrialized world remain to be investigated, and that's also questions that we are focusing now in our lab. So I think I have now completed my presentation. I think I'm on time, and I want to really acknowledge my colleague, Professor Mathieu Groussin, who co-founded the Global Microbiome Conservancy with me, and who's been leading this research, co-leading this research with me. Really thankful to our students here in our labs, as well as the Global Microbiome Conservancy Consortium, all these people as well as sponsors, and thank you for your attention. I'd be very happy to take questions. Thanks.
Thank you, Matthew. We are out of time, but thank you for this interesting presentation. We will